The principles of immunology are widely applied in the development of vaccines and diagnostics of various diseases. Vaccines are biological preparations that contain weakened or dead pathogens or parts of them. Vaccines are biological preparations that contain weakened or dead pathogens or parts of them which stimulate the immune system to produce a protective response against the actual pathogen. Diagnostics, on the other hand, involve the detection and identification of specific pathogens in clinical samples, usually by using specific antibodies. Vaccines are important tools for the prevention and control of infectious diseases. Vaccines are important tools for the prevention and control of infectious diseases. Some of the major examples of vaccines developed using immunological principles include the papillovirus, smallpox vaccine, measles vaccine, and the COVID-19 vaccine. Vaccines have been successful in eradicating diseases such as smallpox and reducing the incidence of diseases such as polio and measles. The use of vaccines has also been instrumental in reducing the burden of infectious diseases in developing countries. So diagnostics play a critical role in early detection and treatment of diseases. The principles of immunology are used in the development of diagnostic tests such as ELISA, enzyme-linked immunosorbent assay, PCR, polymerase chain reaction, and rapid diagnostic tests. These tests can be used to detect a wide range of diseases such as HIV, hepatitis, malaria, and COVID-19. They are highly specific and sensitive and can be used to detect diseases even at very low concentrations. Then tissue and cell culture methods are widely used in the field of biotechnology for the production of small and micromolecules. Tissue culture involves the growth of cells, tissues, and organs in vitro under controlled conditions. Advantages of tissue culture and cell culture methods include the ability to produce large quantities of specific products, the ability to control the growth conditions and the purity of the product, and the ability to modify the cells to produce specific products. Then disadvantages include the high cost of equipment and reagents and need for skilled personnel and the risk of contamination. So in conclusion, the principles of immunology and tissue and cell culture methods have revolutionized the field of biotechnology and have led to the development of vaccines, diagnostics, and a wide range of products. These methods have played a crucial role in the prevention and treatment of diseases and have contributed significantly to the growth of the biotechnology industry. However, there are still challenges such as cost and complexity of these methods that need to be addressed to make these technologies more accessible and affordable to people around the world. Then transgenic animals and plants. Transgenic animals and plants are organisms that have had their genetic material artificially modified by the introduction of foreign genes using molecular biology techniques. These techniques enable scientists to add, delete, or modify genes, resulting in the production of transgenic organisms. The use of transgenic animals and plants has gained popularity in recent years due to their potential applications in medicine, agriculture, and environmental management. One major application of transgenic animals is the production of recombinant proteins used in the treatment of human diseases. For example, transgenic cows are used to produce human proteins such as lactoferrin and antithrombin 3 in the milk. These proteins are then purified and used to treat various medical conditions, including blood clotting disorders and infections. Then, transgenic animals are also used in basic research to study the function of specific genes and the mechanisms of various diseases. For instance, mice that have been genetically modified to develop cancer or other disease are used to study the disease process and test new drugs. Similarly, transgenic plants have significant applications in agriculture, including the development of crops that are resistant to pests, diseases, and herbicides. For example, genetically modified soybeans have been developed to be resistant to herbicides, making them easier to grow and increasing crop yields. Then transgenic plants can also be modified to have higher nutritional content, such as golden rice, which is genetically modified to produce higher levels of vitamin A. This has the potential to alleviate vitamin A deficiency in populations that rely on rice as their primary food source. In addition to transgenic animals and plants, molecular approaches to diagnostic purpose and strain identification has become the essential tool in medical and environmental science. Polymerase chain reaction is a molecular technique used to amplify DNA samples and can be used to detect pathogens, identify genetic disorders, and diagnose various diseases. PCR can also be used in environmental monitoring to detect and quantify microbial populations in soil, water, and air. This enables scientists to identify and monitor potential environmental hazards and assess the efficiency of environmental management strategies. However, the use of transgenic organisms and molecular techniques also raises ethical and safety concerns, like transgenic organisms may have unintended effects on ecosystems and may pose a risk to human health if not carefully regulated. Additionally, the misuse of molecular techniques could have severe consequences, including the release of genetically modified organisms into the environment. In conclusion, transgenic animals and plants and molecular techniques have significant potential applications in medicine, agriculture, and environmental management. They offer numerous advantages, including the production of valuable proteins and crops, disease diagnosis, and environmental monitoring. However, it is essential to ensure that their use is carefully regulated to minimize potential risks to human health and the environment. Then, genomics and related topics. Genomics is the study of an organism's entire genome, including the interactions between genes, the environment, and other factors that contribute to the development of the organism. The field of genomics has revolutionized the way we study and understand the genetic basis of human health and diseases, as well as the genetic diversity of plant and animal species. One of the most significant applications of genomics is in the field of health. Genomics research has led to the identification of numerous genes that contribute to the development of diseases such as cancer, Alzheimer's disease, and diabetes. By identifying the genes involved in these diseases, scientists can develop targeted treatments that can help to prevent or cure these conditions. For example, in the case of cancer, researchers have identified a number of genes that are involved in the development of tumors. By analyzing the genetic makeup of individual tumors, doctors can develop personalized treatment plans that target the specific mutations that are driving the growth of the cancer. This approach, known as precision medicine, has the potential to revolutionize cancer treatment by improving patient outcomes and reducing side effects. So, in the case of cancer, researchers have identified a number of genes that are involved in the development of tumors. By analyzing the genetic makeup of individual tumors, doctors can develop personalized treatment plans that target the specific mutations that are driving the growth of the cancer. This approach, known as precision medicine, has the potential to revolutionize cancer treatment by improving patient outcomes and reducing side effects. In agriculture, genomics is being used to develop new crop varieties that are more resistant to pests and diseases, as well as to improve the nutritional content of the crops. By analyzing the genetic makeup of crops, scientists can identify the genes responsible for desirable traits such as drought resistance, disease resistance, improved yield, etc. For example, scientists have developed a genetically modified rice variety that is enriched with vitamin A nutrient that is essential for human health. This rice variety, known as golden rice, has the potential to help to reduce vitamin A deficiency in developing countries where rice is a staple food. Another application for genomics is gene therapy, which involves the use of genetic material to treat or cure disease. Gene therapy holds great promise for treating a wide range of diseases, including genetic disorders such as cystic fibrosis and sickle cell anemia. However, there are also potential risks associated with gene therapy, including the possibility of unintended effects on the patient's genome. Additionally, there are ethical concerns surrounding the use of gene therapy, particularly in cases where it involves the alteration of the genetic makeup of future generations. In conclusion, genomics has enormous potential for improving human health and agricultural productivity, as well as for advancing our understanding of the genetic basis of life. However, it is important to carefully consider the potential risks and ethical implications of these new technologies and to work to ensure that they are used in ways that benefit society as a whole. Then, biosources. Biosources refer to the living components of nature that can be used for various purposes, such as food, medicine, and industrial products. Biodiversity, on the other hand, refer to the variety of life forms found in ecosystems. The bioresource sector is a crucial aspect of sustainable development as it plays a significant role in maintaining purposes such as food, medicine, and industrial product. Biodiversity, on the other hand, refers to the variety of life forms found in an ecosystem. The bioresource sector is a crucial aspect of sustainable development as it plays a significant role in maintaining the ecological balance and generating economic benefits. So, bioresource refers to the renewable resources derived from living organisms that can be utilized for economic, social, or cultural purposes. These resources include plants, animals, and microorganisms. The bioresource sector is essential for a country's sustainable development as it provides raw materials for industries, contribute to food security, and creates employment opportunities. The utilization of bioresources has been instrumental in the development of several industries such as pharmaceuticals, biotechnology, etc.